right, so this is part 2 of your chapter 4, membrane transport, starting with osmosis. So osmosis is defined as diffusion of water through a membrane. And there are some characteristics that this is a form of passive transport, right? So no energy is put into moving water across the membrane. And water is not affected by membrane potential, so the voltage on the membrane does not matter. Water depends on actually the solute concentrations inside and outside the cell. And it's driven by a water gradient, which is basically just a concentration gradient of water, right? So now instead of looking at the solutes or the molecules dissolved in the water, we're looking at water itself. So there's a term called osmotic pressure, which is the net flux of water into or out of the cell. And so we actually want our cells to be in a equilibrium environment where there is no net flux of water in or out of the cell. We want our cells to be in what's called an isotonic or isoosmotic environment. But if you change the environment that the cell is in, you can change the flux of water, and that can cause problems for living organisms. So the pressure is the fact that you can actually force the water to diffuse in one direction or another. And solutes are the substances that are going to control whether or not water moves. So where water goes where the solutes go. So the solutes are going to move first, right? You're going to change the solute concentration either inside or outside the cell, and then the water will follow. So osmotic pressure is a pressure that would have to be applied to stop the water moving according to its concentration gradient. So let's look at this couple of terms. So in the outline, we go through osmolarity and tonicity, but we're going to spend more time on tonicity. Remember that osmosis, water is moving through proteins called aquaporins, so it's a form of facilitated diffusion. Aquaporins are always open, they're not gated. And so looking at osmolarity, so the normal solute concentration inside the cell is around 300 milliosmoles. So the measurement, the unit is milliosmoles. And if you put the cell into an environment where there's the same milliosmolar concentration outside as inside the cell, and that's an isoosmotic solution, and water is going to have no net flux between the inside and outside. If you look at a hyperosmotic versus hypoosmotic solution, this is where we have some flux of water. So if we take a look at our picture here, right, the hyperosmotic side is going to have a higher solute concentration and therefore a lower free water concentration. The hypoosmotic side will have a lower solute concentration and therefore a higher free water concentration. So looking at water only, right, let's just name these side A and side B. Side B has a higher water concentration. The water concentration is higher. So water will flow down its concentration gradient, right, towards the opposite side. So water will flow from side B towards side A. And that's the osmotic pressure. So the hyperosmotic side, the side with more solutes in it, will be the side where water wants to run to, because water wants to reach equilibrium. And so it wants the concentrations of both sides to be the same. And so the water will move, increasing the water content in A until equilibrium has been reached. Okay, so that is osmolarity. And osmolarity, these solutes in osmolarity, it's for looking at osmolarity, we are looking at the total amount of permeating solutes and non-permeating solutes. So maybe these solutes can cross over to the other side, maybe they can't. So it's the total number of solutes we're looking at when we talk about osmolarity. When we talk about tonicity, then this is going to be looking at only non-permeating solutes. So these solutes cannot move across the cell membrane. They're trapped on either side of the cell membrane. And what will happen is that if there's an imbalance of tonicity from the inside and the outside of the cell, then the cell will either shrink or swell and change its volume to try and equalize the concentration gradient of water on either side. And so we'll just look at a couple of examples here. So the volume of the cell changes if the tonicity is changing. So if we look at this example here, so 300 milliosmoles is what's inside the cell, and we have 150 milliosmoles outside the cell. So in this kind of example, we have the cell is in a hypotonic environment. And the word, when we use hypotonic, hypertonic, and isotonic, we're always referring to what the fluid is outside the cell. So these environments are always talking about the extracellular fluid. Okay, so because the cells will always constantly have 300 milliosmoles as their, as kind of like their baseline normal physiological solute concentration. So if a cell is placed in a hypotonic environment, the pressure, right, the water is going to want to move towards the hypertonic side. In this case, the hypertonic side would be inside the cell because it wants to balance out the concentration of water. The outside of the cell here is more concentrated in water, less concentrated in solute, right? It's hypotonic, less solute. So water will enter into the cell until when? Until the concentration gradients are equalized. And that means that since we had half of the solute concentration outside, the cell will have to double in size in order to reach an equal concentration of osmolarity. If we look at another example here where we have a cell inside of a hypertonic environment, okay, then water will move outside of the cell. And how much water will move outside the cell? Well, until the osmolarities are equal. And for that to happen, since the outside is twice the osmolarity as inside, we have to shrink by half in order to equalize the concentration gradient or the osmolarity. So the cell, the overall lesson here is that water moves according to its concentration gradient, and that concentration gradient is set up by the solutes. So wherever there is more solutes, that's where you have less water concentrated. Water will run from high to low concentration towards the hypertonic side until it reaches equilibrium. In order for equilibrium to be reached, right, when water moves, the cell will either swell or the cell will shrink in size, changing the volume. And the volume change will be proportional to the difference in the tonicity. Okay. So let's see if we can answer this question. What happens to a cell that's put into a hypertonic solution? Okay, so if we think about the hypertonic solution, that means outside the cell, it has more solutes. Water always moves in the direction of where there is more solutes. Water will leave the cell and the cell will shrink. All right, so this brings us to this notion of dehydration versus water intoxication. So let's just take a look at what's going on here with this first. This is dehydration, and the second scenario here is water intoxication. So here's our intracellular fluid inside the cells. We know this is 300 milliosmoles, right? Beginning off anyways. So the extracellular fluid, when you are sweating and dehydrating, what's happening is you're losing water from the extracellular fluid, and your solute concentration will begin to concentrate, right? So we're not taking in any water. We're not in this scenario. We're just sweating, and we're not taking in any extra fluids. So what happens here is that the solute concentration in our extracellular fluid will start to concentrate, right? Because we're losing that water, and this is going to create a hypertonic environment. Water will leave our cells and get sort of moved outside into the extracellular fluid, and that's going to cause our cells to shrink a little bit. right? So water leaves the intracellular fluid by osmosis. If we look at the second scenario, we have a situation called water intoxication, where someone is drinking an excess amount of water. So you're drinking copious amounts of water. What this leads to is that you are adding water and you're diluting the solute concentration. So the solute concentration of the extracellular fluid decreases, right? So compared to inside the cell now, the inside of the cell is going to have more solutes compared to the outside because you diluted it by drinking so much water. So water is going to move through osmosis into the cells, and cells will start to swell. So let's do this table, this exercise, in talking about changes in body fluid compartments. So this all works on the idea of osmosis. So in our first condition, what if a person is placed on an IV of isotonic saline? So when you go to the hospital and you get fluids, they don't put just pure water into your body. They're putting an isotonic saline solution. So what happens to your total body water? Well, of course, the total body water, how much water is in your body, will increase, right? Because you're putting the saline into your water. The extracellular fluid concentration is going to go up. Okay? And what's the effect on osmolarity is that it's the same. There's no change because we're putting in an isotonic solution. So we're not changing the tonicity or the osmolarity. There's no extra solutes or less solutes. Because we're putting in, all we're doing is we're increasing the volume of the fluid outside of our cells, but since it's isotonic, the cells are not going to increase in, the cells themselves will not take up water or lose water because it's isotonic. So for intracellular concentration, intracellular fluid, there's no change because it's, again, isotonic. What about diarrhea, where you have isotonic loss? Okay, so overall, we know that total body water will decrease, right? Because we have diarrhea, water is leaving our bodies. So that's going to create this extracellular fluid to decrease. Inside the cells, though, there's still no change because we're losing an isotonic fluid, all right? So there's no change into the osmolarity because we're not losing just water, we're losing this isotonic fluid. So both the solutes and the water are being lost, but in terms of the relationship between intracellular and extracellular, there's no change. The osmolarity, there's no change in osmolarity. Let's look at the third example here where we have excessive salt intake. So this is like eating some really salty potato chips. You just ate a ginormous bag of potato chips. You did not drink any water while you're increasing the salt content. So what happens is the total body water that you have is the same, right? There's no change here because all we're doing is we're adding salt. We're not adding any water. So the total body water is the same. The extracellular fluid and intracellular fluid, well, let's just take a look at that for a second. So what is going to increase the osmolarity? So this is going to increase the osmolarity because we're adding solutes. We're adding salt, okay? And we're adding solutes that actually are not permeating. Salt does not permeate. So what happens to the relationship in our intracellular fluid and our extracellular fluid? So the extracellular fluid volume, we know that water will go to the salt, right? If you're adding salt into the extracellular fluid, then water will follow. So the extracellular fluid will go up. But where is that fluid coming from? Where is the water coming from? It's coming from inside the cells. So the intracellular fluid will decrease. All right, now let's look at excessive sweating, hypotonic loss. So we're losing just water, right? We're losing water, we're not losing electrolytes. So what happens to our total body water? It's going to decrease, right? What happens to our osmolarity? Our osmolarity will also increase because if we lose water, we're going to concentrate our solutes, right? Because we're losing the water part, we're concentrating our solutes. So really, this scenario and this scenario is the same. So if we're concentrating our solutes on the outside of our cells, the extracellular fluid will increase, right? Because again, water moves to the side of the cell where there's more solutes. And where is the water coming from? It's coming from inside of our cells, the intracellular fluid, and so that volume will also decrease. So I hope this table makes sense to you, and we can take a look at the next topic here, active transport. So active transport is non-spontaneous, which means that it is going to not go down the concentration gradient, but actually go up the concentration gradient. Excuse me. So the difference between active and passive is that we're moving molecules up their concentration gradient instead of down. This requires energy, and it requires a transmembrane pump. So it's going to require protein. Sometimes we call them pumps. So these pumps are going to function as a transporter and an enzyme. Why an enzyme? Because it hydrolyzes ATP. So a lot of time, these transporters, when we talk about the sodium-potassium pump in the nervous system, sometimes they're also called ATP ACEs because they break down ATP. So they're able to harness energy. They have, again, specific binding sites, so they're specific. And they also can hit a saturation point. So, the overall is that the movement is uphill. All right, so let's take a look at the sodium-potassium pump, which is something that we're going to look at a lot more in the nervous system. So this is going to be primary active transport. Okay, so what the difference between primary and secondary is, primary active transport directly uses ATP. So it functions as an ATP ACE. We can see that ATP is being used. It actually binds to the transporter protein and it becomes phosphorylated. And then after it functions, the phosphorylation, right, you lose that phosphorylation and we can go back to the normal conformation. So walking through the function of the sodium-potassium pump, we can see that there are binding sites for sodium ions on the intracellular side, and it's going to bind the sodium ions. Binding will also change the conformation of the protein slightly. Binding ATP is also going to change the conformation of the protein. So notice that ATP is hydrolyzed. The phosphorylation of the pump allows it to change conformation and turn its binding sites outwards. And when it does that, it decreases the affinity for sodium and increases the affinity for potassium. So one little detail to look for here is that these binding sites, the shapes change, right? So intracellularly, when the pump faces inside, it has a high affinity for sodium and a low affinity for potassium. The binding and hydrolyzing ATP causes this conformation change so that the sodiums are released extracellularly. And we can see that the affinity has changed, right? So now the potassium binding sites, there's more affinity for potassium and there's less affinity for sodium. So then the potassium will bind in this picture, right? And then the release of the phosphate group changes the conformation again, releasing the potassium intracellularly by changing that binding site. So it's less affinity for potassium, potassium is released, and then we're back to that high affinity for sodium. So the sodium-potassium pump constantly works to move both sodium and potassium against their concentration gradients, directly using the energy stored in ATP. So due to this pump, the intracellular concentration of potassium, right, since we're moving potassium inside the cell against this concentration gradient, we're going to have a very high concentration of potassium inside the cell, and we have a lower concentration of potassium outside. And then for sodium, we have a higher concentration of sodium outside the cell. Oops, just 1 plus, there's no 2 plus there. There's a higher concentration of sodium outside the cell, but the cell constantly works to create this concentration gradient. So we're moving sodium outside towards its higher concentration gradient, moving potassium inside towards its higher concentration gradient. So the cell is setting up a difference in concentration of potassium and sodium. All right. Other examples of this are going to be proton potassium pump in the stomach. We'll look at that in the digestive system, and then calcium pumps in the muscles. Let's look at secondary active transport. So what secondary active transport does is it uses the energy within a concentration gradient. So it's not directly powered by ATP. However, it is active because ATP is used to set up a concentration gradient, and then the secondary active transport uses that concentration gradient as potential energy to move substances against their concentration gradient, right? So energy is released from an ion diffusion gradient energy drives the pump, right? So we're not directly using ATP, we're using a concentration gradient. Diffusion results from previous active transport of the ion, right? So previously we have used ATP to create this concentration gradient, but in secondary active transport, we're using this potential energy stored in that concentration gradient to move. So let's talk about the two kinds here. There's co-transport or symport, where we're moving the substances in one direction, and then counter-transport or antiport, where we're moving substances in different directions. So this is an example of co-transport. Co-transport or symport, you're moving two substances in the same direction. However, notice that sodium, we just talked about how sodium is very high concentration outside the cell, and it's a lower concentration inside the cell, right? And so sodium is moving down its concentration gradient here. And the transporter protein here can use this potential energy released in moving sodium down its concentration gradient to also move glucose up its concentration gradient. So glucose is going to be at a lower concentration outside the cell, right? The cell wants to eat glucose, so glucose concentration is higher inside the cell, but we want to move more glucose in the cell, so we're moving it against its concentration gradient. Right? So sodium moves down the concentration gradient, and the transporter is going to use this release in potential energy as sodium moves to help glucose be transported against its concentration gradient. However, the idea is that both sodium and glucose are moving in the same direction, which is why it's called symport or co-transport. And this is an image of countertransport. So it's the same idea where we're using sodium's flowing down its concentration gradient, right? We're releasing the potential energy in that action, but we're just moving the hydrogen ions in the opposite direction, but still against its concentration gradient, right? So sodium is moving from high to low. This is down the concentration gradient. But here, the hydrogen ions are going to move towards a higher concentration gradient, these protons, compared to inside the cell where there's a lower concentration of protons. So it's moving against its concentration gradient, but, using that potential energy in sodium movement, and it's called countertransport because sodium and the proton or the hydrogen ion are moving in different directions. All right, so these are just a wrap-up of different kinds of transport. We have simple diffusion. So we have, and then we have sort of mediated transport. So simple diffusion, remember, is where we're just moving substances through the membrane bilayer, and the motion of movement depends on whether those items are hydrophobic or nonpolar. There's no saturation, there's no specificity, there's no energy source, right? There's no transport protein required, and things go down their electrochemical gradient. Then when we look at mediated transport, right, this is where you need a protein. So if it's mediated, you just need a protein to help. And these can be passive or active. So passive transport, we have channels and we have facilitated diffusion, right? Both of these, since it's passive, they're both moving down their electrochemical gradient. Yes, there's transporter proteins required. In facilitated diffusion, they're called carriers. In passive transport, for channels, right, they're called channels. Does it require energy? No, because this is passive movement. Sometimes channel proteins can get saturated, but we looked at how facilitated diffusion becomes saturated. Is there specificity? Yes, right? Channels and facilitated diffusion, those proteins, anytime you have a protein, it's specific. And the characteristics of these things, sorry about that. So channels are usually put in place, and facilitated diffusion as well are put in place to move substances that don't usually cross the plasma membrane. So hydrophilic substances, either they're ionized or they're polar. And these are some examples of things that move through channels and things that move through facilitated diffusion. So channel proteins, any ion, they move through ion channels. And then we look at active transport. So now we're moving up an electrochemical gradient. We must use a transport protein, a pump. Yes, we require energy. In primary active transport, we're using directly ATP. In secondary active transport, we're using an electrochemical gradient to help another solute move against their concentration gradient. Both of these can be saturated, they're specific, and what kinds of things are moving through. Again, if things that cannot pass through the membrane on their own, so hydrophilic molecules are pumped past the membrane, and then primary active transport is usually for inorganic ions, and secondary is usually for glucose or amino acids, but you can also see we looked at protons and calcium ions. The examples are not as important, so don't worry about examples, but all the rest of these should make sense to you. Okay, so let's talk about steady state. So a steady state is present because leaks across the membrane are occurring passively at a slow rate. So this is coming back to the sodium-potassium pump. I should have just talked about this a little bit before. Coming back to the sodium-potassium pump, like why does the pump always work? How come the pump just doesn't pump the cell completely dry of sodium and then pumps all the potassium from outside in? It continuously has to work because there are also some leaks, channels in the membrane. So you can see that sodium does leak in through these channel proteins, and potassium does leak out. So they leak according to their electrochemical gradient, the direction that they're moving. But the pump has to, so that's why the pump has to work constantly to maintain the the different concentration gradients from inside the cell and outside the cell. All right, so 5 concept questions to review. Take a look at this first one, and what's the answer? All right, so chemical driving force. So just looking at the chemical driving force, which tends to cause molecules to move in a particular direction, is a direct result of which of the following? So it's going to be the concentration gradient of the molecules. The name for the term that refers to the number of molecules that cross a membrane per unit time, per unit membrane surface area is its flux. Which of the following factors does not influence the rate of simple diffusion of molecules across the membrane? All right, so we're talking about diffusion, so the only thing that's not going to is the active transport, right? So permeability is one of those factors. The magnitude of the driving force, how big is the concentration gradient? Are we talking about a little gradient or a big gradient? Or I should draw it more like a flatter triangle versus more steep triangle. And the surface area does. So letter D is the answer there. In the sodium proton exchange, energy released when sodium flows down its concentration gradient into a cell is used to drive hydrogen ions out against its concentration gradient. What is this an example of? So this is going to be our secondary active transport because we're using a concentration gradient. And is it going to be, so we can cross out our primary, that's not it. So is it secondary active transport and counter transport? This is our answer. Which of the following does not influence the rate of a facilitated fusion of molecules across the membrane? So since we're using facilitated diffusion, this is going to use a carrier, right, molecule that's going to help transport these molecules. So we don't need to worry about the solubility of the molecules in the membrane. That's not going to be a factor. Okay, so answer D is your answer to this question. Individual carriers, the transport rate is a factor. The number of carriers is a factor. And then just the force, the electrochemical force and gradient is also a factor because, again, we're talking about diffusion. All right, so that's the end of part 2.